been tripping and I might go crazy since I've been in flight mode.
Okay, well, this is a great surprise, I must say. It's amazing to be back in Amsterdam. This is where I studied biology, and some people from those days are on the first row here. It's great to be back in this beautiful city that we just heard a song about. And what I will do today in the next 45 minutes or so, I want to talk about genetic modification. And the title of this presentation has been made up by the organization of this Cocktails in Science, genetically modifying everything. Well, honestly, I don't think so. What I want to share with you today is I will talk about the CRISPR-Cas system, and of course that is well known because of what we can use it for. So that's the genome editing. But what I want to share with you tonight is to discuss what is possible and what do we want, because a lot is possible, as you will see, but some things we certainly do not want. Okay, before I start the presentation, this is what these days is asked from my employer, Wageningen University, to give a disclosure. I work at several companies, as you can see, involved in gene therapy, pathogen diagnostics, and crop improvement. And also, I together with some colleagues, we have several patents which are owned by the university. And the ongoing projects are funded by NWO, the ERC, the EU, and some private sponsors. And one of those private sponsors I will come back to at the very end of the presentation. Okay, so this is what we are going to discuss tonight. So these are the topics. So first we start with DNA, very briefly. Then we turn to CRISPR. I want to explain to you at least the basic details that you need to know to understand what indeed we can do, what are the possibilities. And then I will give you some spectacular examples. But then I will also want to bring up a dilemma. So how far do we want to go? Where do we draw the line? And then at the end, a surprise act. Okay, so about DNA. So what you probably all know is that DNA is responsible for storing the genetic information. DNA is based, DNA is basically making up the chromosomes that occur in all living organisms on this planet. So from bacteria to plants to humans. And even in a lot of viruses, DNA is the genetic material. And so that also means that it occurs in every living cell. So of course, a bacterium is a single cellular organism, has a single chromosome. We have a lot of cells, billions of them, and each of those cells, they have DNA. That's important to know. Then, who discovered the basic features of the DNA? Well, it was these two guys, Watson and Crick, and it was exactly 70 years ago that they published this paper in Nature, and I will blow up the figure. So this is the drawing they included in this milestone paper with the double helix. What you do not see here, and that's very important, is that the two strands of the DNA are held together because of base pairing. So, for instance, the yellow and the purple, that's one combination of bases. For instance, it's the GC base pairing, whereas the orange and green, that's the AT. And they come in different orientations, that doesn't matter, but that specific interaction is key for the functionality of the DNA molecule. And that's, I mean, if you have time, you should look up this paper. It's really a piece of literature. And clearly it has been, Jim Watson was a guy from the United States. Francis Crick was a typical Englishman, and I'm sure he wrote these sentences. So this structure has novel features which are of considerable biological interest. It has not escaped our notice that the specific pairing we have postulated suggests a possible copying mechanism for the genetic material. And that was, in fact, the next big step these guys made. And it was in a paper that was, so it was basically the 15 years after this discovery of the double helix that they figured out a lot of details of the molecular biology. And that was summarized in this paper from 1970, again by Crick. And what he launched here was this cartoon. And that's really kind of summarizing all the big or the important details of the molecular biology. He called it the central dogma. And what he states here is that basically there is a flow of information from one polymer molecule to another. So DNA is a polymer. So I already showed you the basic components, GATC, but that has to be translated all the way via RNA to protein. And proteins have completely different building blocks. But also, as is indicated on the top, DNA is going to replicate, because every dividing cell, before every cell division, the DNA has to be replicated. So how does that work? So let's focus on this DNA replication mechanism, and then you see that because of that specific interaction, there is, if the two strands are separated, there is only one way to fit in the building blocks of the second strand. And that's true for the one strand, but also for the other strand. And then you get two exact copies of the two DNA molecules. Okay, and then let's go one step further, because then the information that is stored in the genetic material, it has to be expressed, right? Because DNA does not have all the functions that are present in our living cells. So, but proteins do. Okay, so but the information stored in the DNA then has to be transcribed to RNA, which is kind of a similar molecule as DNA, as we will see. And then it has to be translated to completely other building blocks to proteins. How does that work? Okay, again, the two strands have to be separated, and then one of the DNA strands forms the template of an RNA molecule. Okay, and it's called a messenger RNA molecule because it contains the message of the gene that has been expressed. So this is the RNA molecule, and then there has to be a translation step. So then you have to, basically the sequence of the building blocks from DNA and RNA, it has to be translated to the building blocks of proteins. And that big yellow blob, that's one of the building blocks, the first building block of the protein. And as you can see, it's another RNA molecule, a transfer RNA, that is responsible for delivery of those, the specific delivery of specific building blocks of the protein. So here the second one comes in, then a connection, a peptide bond is being formed, the link with the RNA is being broken, then the next step or the next amino acid comes in, and so on. And that, of course, this is only 3 amino acids, and proteins can exist of 100 to 1,000 of even more. But this is just an example to illustrate how that works. So now we know how we can use the information on the DNA to generate the functional units, and those are often the proteins. Okay, so to wrap up this first introduction part, so we know that the DNA carries the genetic information, but what we now also learned is that the DNA has to be copied, as the cell divides. Then, unfortunately, during that copy process, although it's very accurate, still mistakes are being introduced. In humans, for instance, it's 1 in every 10 million or every 100 million duplication events that mistakes are being introduced. In viruses, we know that the mutation rate is much higher. And that was also one of the problems of that we experienced during the COVID pandemic, right? Those viruses, when they replicate, they introduce a lot of mutations, so they kind of find a way around the vaccines that were developed. Okay, some other features is, of course, it's when something like this happens in a replicating human cell, that can have devastating effects. It can, if the mutation is in a certain gene at a certain location within that gene, it can give rise to genetic disease, because then a different amino acid, for instance, is being incorporated. That's what we now know how that works, right? So one letter different at the DNA level can give rise to one of the building blocks that is missing or that's different in a protein, and when that's on a crucial position, that can have really bad consequences. Of course, there is also a good thing, because if there would not have been any mistakes during replication, during evolution, there would be no evolution. And then we would not be here, right? Then we would all still be bacteria crawling in the mud somewhere. But we are not. So that's also one of the good things we can say. Well, it depends who you ask, but it's one of the good things. Let's stick to that. Okay, then about CRISPR-Cas. So CRISPR stands for clustered, regularly interspaced short palindromic repeats. And the 2 important words here are the spacers and the repeats, because they are located in such a CRISPR locus that you see up here. In black, the repeats, they are exact copies of something like 30 base pairs, 30 letters, and then in between, of a similar size, more or less, you have the so-called spacers, and those spacers are different each time. Okay, so and then next to those CRISPR arrays on the chromosome, this is the chromosome of a bacterial cell that you see over here, and next to those CRISPR arrays, you find the genes that code for the proteins that are very important in this CRISPR system. So they are called the CRISPR-associated genes coding for the Cas genes coding for the Cas proteins. Okay, and what I also should tell you is that this CRISPR system, it occurs in half of the bacteria that we know, and what it does is it's a defense system against viruses. So just like we have problems with viruses, and plants also have problems with viruses, also bacteria have their own viruses. And they, just like us, also these bacteria have developed or have evolved defense systems. And this CRISPR system is one of the most sophisticated defense systems that were discovered, and it was discovered some 15 years ago. So here you see an infection of a so-called bacteriophage, so that's a bacterial virus, and it introduces its genetic material inside the bacterium, inside the bacterial cell, And then it can happen that when all goes well for the virus, then the virus replicates or duplicates in say 20 minutes or so, when all the conditions are perfect. And then so something like 50 or 100 viral particles are being generated, and that kills the cell. But when the conditions are not that good, or when, for instance, this virus has a mutation and it cannot replicate that fast anymore, then the CRISPR system can act. And what it does then, it selects a fragment of the incoming viral DNA, and it incorporates that as a new part of the CRISPR array. So it's like the CRISPR array is like the memory of the immune system. Okay. Then again, as we said, well, this is just storing information, but it has to be transcribed. So it is being transcribed as one big RNA molecule, and that molecule is then being processed, and that eventually, this type of RNA is not being translated, so no protein is being formed. Okay, so the CRISPR RNA, that's the final stage, right, of the RNA, and that is very important. But the genes, the Cas genes, they are being transcribed, but also translated. So they end up as proteins, right? The Cas proteins, and some of these Cas proteins can bind the RNA molecule, and the RNA molecules, they are called the guides, the CRISPR guides, because they guide the DNA targeting Cas genes or Cas proteins. They guide them to a target sequence. And there, it's very important that we now know how this base pairing works, because then you can, if this guide indeed finds a matching DNA molecule, then it can, through the base pairing, exactly what I just told you, through that base pairing, it can find a target molecule, so in this case a viral DNA, and it can cut the DNA and kind of neutralize the viral infection. Okay, let's now zoom in on this targeting event. And first of all, this is what you see is kind of happening. So in the middle, you see the RNA, the CRISPR RNA molecule. These are all Cas proteins, and we gave this complex the name cascade for CRISPR-associated complex for antiviral defense. And then it starts to find a matching DNA sequence that matches with this RNA guide. Well, in this case, that was not a good match, but in the second time, you will see that there you find a match, and then one of the DNA strands is base pairing with the guide over here, and the other strand is being displaced. And then, because of this structural rearrangement, a last nuclease Cas protein enters the scene, and that starts to damage the DNA. So here you see that this system works perfectly as a system to very specifically target viruses. And here again, so using the same cartoon, here you can see that, or how that works. So here you see again the Cas protein with the guide molecule. And here you see a target, and then what we saw in the movie, you see, or you get the displacement of one strand, and then you get stable binding of the CRISPR guide with one of the DNA strands, and then this nuclease can, as the name suggests, it can cut the DNA very specifically. And that's what you see here with the red arrowheads, that those strands are being cleaved very specifically. So only this part of the viral DNA is being targeted, and that's very good to remember also for the applications that we will talk about in a minute. And it's also good to realize that in this example, I used a guide of only 9 letters, and that's of course way too short to get a unique cut in the human DNA. The human DNA altogether is something like 3 billion base pairs. So that's a lot. And if you would use a guide like this, that wouldn't work, because that sequence on average occurs 10,000 times in the genome. So you would cut the whole genome, and that's destroying the cell completely. The good thing is that in nature, the natural bacterial CRISPR systems, they typically use guides of say, 20 letters. And this particular word, or a particular word of the 4 letters, GATC, occurs, well, you can do the math, it's 4 to the power 20, that's a lot. And if you then calculate how many times this would find a matching sequence in the DNA of humans, that is only once on average. Okay? So using this system, we can then very specifically find a particular DNA sequence on the human genome, or on the plant genome, or of a bacterial genome. It works on any genome. And it's basically, it compares to the Control F function on your computer. So if you type in this certain word and you look for it in a text file, you will find a couple of hits. But the longer the word, the less likely it will be that you find it. Okay, so this really allows for genome editing. And what we saw in the movie was kind of this. We had the RNA molecule held by this CRISPR complex, and then this other protein was recruited, and then it was basically destroying one of the strands. Now that's perfect for a bacterial defense system against viruses, but it's not perfect for genome editing, because then you want to have a precise cut, and then at that location you want to do the editing. You want to repair it at that level. And then later on, other Cas proteins were also characterized. So in 2012, the Cas9 was characterized, and there, as you can see, that system gives very nice cuts of the strand. So it does not destroy a complete strand as the cascade complex, but it gives very nice cuts of the 2 strands. And also this type 5 system, a Cas12, we discovered that in 2015, and also that gives rather nice cuts of the 2 DNA strands. So these systems can be very nicely used for editing, but this one is kind of not well suited for that purpose. Okay, then suppose we want to repair a certain mutation at the DNA of whatever creature. So the mistake that we want to repair is located at this particular sequence. So we design a guide, we order them, and these days, in a couple of days, you have your guide, and then you can, together with the protein that you have to produce yourself, but that's also not rocket science. You can then, together, the protein together with the guide, you can introduce that in a cell that you want to do the editing, and then because of the specificity of the guide, it will find this sequence, and it will, very close to that particular sequence where you want to do the edit, you can generate a double-strand break. And then that's basically where the task of the CRISPR system stops. Some people refer to CRISPR-Cas as cut and paste, but it only cuts. And the paste, so the repair actually has to come from another system, and those systems are the native repair systems of the cell that you want to edit. Okay. And then there are roughly two types of repair systems. One is not based on homology, so it's kind of a quick and dirty stitching together of the two ends of the chromosome, and that often leads to some insertions or deletions, so mutations in the DNA. And when that occurs in a gene, it often leads to inactivation of a gene. And that's a very useful trick, also for some applications that I will discuss later. So this is a very convenient trick, but if you want to do precision repair, then you really need the other system, and that's the homologous repair system. For that, you need a so-called repair fragment. So together with the Cas protein and the guide, you also have to then introduce in the cell that you want to edit, you have to introduce a DNA with the flanks of the broken chromosome. Okay, so here you get, because of this repair system, it uses this to fix the break. But we can cheat, and the cheating is the green part here, because there you can introduce the edit that you want, and it can be a single letter of the DNA or a single base pair, right? It can also be a couple of base pairs. It can be an entire gene, okay? But the flanks have to be homologous. Then this repair system can do the job, and then you can get very accurate engineering, as is indicated. Okay, some recent examples. This is the Cas9 protein. So Cas9 is probably the most popular. Well, first of all, because of the very nice features of this particular system, but also because it was the first to be completely characterized. And the Cas12 system that I explained, that came only 3 years later, but still it has to catch up with Cas9. Why do I keep on bringing up Cas12? Because it feels like my baby. Because we were we saw the birth of this particular system, so it's close to my heart, and also we have a patent on that. So that also. Okay, so this is an example of editing in human cells. This is I wrote down it's being done ex vivo. That means outside the living body. And in this case, that's what a lot of research currently is going on blood problems. So genetic diseases with blood cells. And this is an example called sickle cell disease, in Dutch sickle cell anemia. That's a single amino acid change on the gene of hemoglobin. And because of that mutation at the DNA level, there is a different amino acids in one of the subunits of the hemoglobin. And that's the result of that single mutation is that the efficiency of the hemoglobin is dramatically reduced, and maybe you know hemoglobin is present in the red blood cells, and it's responsible for the transfer of oxygen through our body. So you can imagine when that does not function properly, that there is a problem. But with CRISPR-Cas, this has been repaired, and you can do it in different ways. You can use the homologous repair system, so the very accurate system, and there you can really edit the single letter at the DNA level or the single base pair at the DNA level, and you can repair it, and then you can get healthy red blood cells here again. You can also do another trick, and that's maybe less risky, because there are some drawbacks for the homologous repair system, and there you can kind of make a knockout of a gene that is responsible for the repression. This is very complicated. Okay, you make a knockout of a gene, and the result is that another type of another hemoglobin gene is then being expressed. That's the hemoglobin gene that only is present or is important at the level of the embryo, and after that, after birth, that's repressed. But if you can target that repression system with CRISPR, you can produce again or induce again the production of this other hemoglobin gene, and that at least partially repairs then the system and partially restores the proper functioning. In vivo, so in the body of a patient, that's a different story. That's much more difficult because there you have to. Oh no, let me finish the blood system, because that is what you do in a test tube, right? So you can do the editing in the test tube, you can do the checking in the test tube, you can select basically the one that worked out well, and then grow that up again. So those are stem cells that we are talking about. You can put them back again in the bone marrow of the patient, and then there are some first examples that people were treated completely as was designed. So that's really fabulous, I would say. So, but of course, you cannot do that with every type of cell. And for instance, the Duchenne disease, so that's a muscle disease. Then, if you want to repair that, you cannot do that with this ex vivo approach. So then you have to do it in vivo, meaning that you have to introduce the CRISPR-Cas system with a specific guide in the patient, in the body, right? And then you have to assume that all the muscle cells get repaired, and that no other damage is occurring, and you cannot check that, and that's why it will take a while to do these in vivo applications. So I think so. One of the in the movie before the talk, it was said, is it a promising technique? Yeah, for sure, it's very promising. But do we promise too much? Well, we should be very careful there. We should not promise too much, because I mean, I gave a talk in Groningen at the beginning of the year, or beginning of last year. And there was after the talk, there was a Duchenne patient, and he asked a question, can you help me? Right, and we cannot help him now. We are working together with a group in Leiden to make steps there, and the systems get more and more specific, but it's still a long way to go. So we should be careful in not promising too much. And then another trick, but that's again. Do we want that? Is it ethical acceptable? Is editing of the embryo, in fact? The good thing would be, in theory, that then you could. So if parents ask for such a treatment because they know that they carry a certain mutation, they carry a disease, then it could be done in theory, and some examples have been published that indeed with the fertilized egg cell, the editing works, right? But and then again you can do a check when I think when the embryo is only 8 cells, you can just take off 1 cell and analyze it. And then the embryo will develop as a normal embryo, no problem. And then you can do a check. So in principle, there are possibilities there, because the good thing, if you do a proper edit, and of course you should make sure that no unwanted changes have been introduced, then you could, this would lead to a healthy kid. And also, it would not be a carrier of the disease anymore. So, I mean, there are interesting options there, but again, this is something that we have altogether, we have to decide that. Do we want that? Okay, some examples. This was the first example in the Netherlands. I must confess I do not know all the details, but it was in fact in Amsterdam they did this, and they kind of used the CRISPR system to influence the expression of some gene, and by doing so, they got a curing or at least improvement of the situation. This is the sickle cell story. Again, this is a company that we interact with every now and then, and they use so most of these stories are dealing with Cas9, but this is an example where the Cas12 system has been used. And they are now in clinical trials, and so hopefully, at least for this particular disease, there is in the nearer future there will be some good news. This is from December, just before Christmas. This was released on the site of the BBC. That this girl, she lives in England, and she suffered from this very nasty leukemia problem, and by again they could take blood cells, T cells in this case, from the body, and they could do the editing. And in this case, it was not a single edit, but it was 3 different edits. And I should talk to some experts in the audience to explain me what exactly they did. But I mean, this is a beautiful example where they could reintroduce this in this girl, in the body of this girl, and she was cured, or at least she's free of cancer as of now. Of course, we have to hope that it will be a stable situation, but this is very, very promising. Okay, then about plants, as I said, DNA is DNA, so if it works for humans, in theory, it also works for plants, and it does. So many, as I will show you in the next couple of slides, many examples have already been released, hundreds of them, in which different types of plants have been edited. But in many cases, these are fundamental studies. But there was one example which was released last year, the end of last year, a couple of months ago, where a group in Japan, or a company in Japan, introduced a tomato in which they again, by making a simple change, so by using this non-homologous system, just a few base pairs changed, they could disrupt a gene, and by doing so, they could enhance the production of this particular compound, GABA, and GABA has been claimed to lower the consumer's blood pressure. If that's true, I don't know. There is some discussion about this, but this is the first example that a CRISPR plant enters the market. Okay, then, very important, where do we draw the line? How far do we want to go? Well, I think or at least if you ask me, it's clear. Of course, when we can use the technique to help people with a genetic disease, we should try to do everything we can. And of course, we should not promise too much, especially for simple diseases, and as of now, specifically blood diseases. There are very promising results, and I think there something will happen in the near future. But so especially the ex vivo applications, that is going to take off in the near future. The other, so inside the body, as discussed, so the Duchenne example, or in embryos, well, we have to see if that is going to be worked on. Then there are also some other potential examples. One is the designer babies. Every time when I talk to a reporter, after 5 minutes, he asks the question, what about designer babies? Well, I'm very much against it. I think it's stupid to do the editing for that particular purpose, to create beautiful babies. I always tell the reporter, when people really think that's very important, that they want to have a kid with the cute looks or whatever, they should think about that when they select their partner. Then about gene doping, so that's for sports. For instance, also there were some stories that maybe CRISPR could help to finally let the Dutch win the world championship. But I think that's not a good idea. Let's do it in a different way. Yeah, so this is all about ethics. So this is where we can draw the line altogether. So we have to see what the politicians, what the political parties, what they want, where they want to draw the line, and then you can try to influence that to some extent. About ethics, I once gave a talk also in Amsterdam, and then there was this artist of Fokke and Sukke. I don't know if you know him. And he during my talk and during the talk of all the speakers, he made a drawing, and so also a drawing about CRISPR. So they say that that they use. Well, I don't have to explain this, I think. So to avoid ethical issues, these guys used the CRISPR, or they tested their own body, but well. Okay, let's then the dilemma about the plant editing, because that's really a big discussion. And I mean, I've been on this stage 4 years ago, and then after the talk, I mean, I was really impressed by all the attacks that I got. Because I was the preacher. It was on a Sunday morning. I was the preacher for CRISPR. But then some people in the audience, they really, they started a very fierce discussion. So in the meantime, I hoped that I'm prepared better for that now. Let's see. What I did now, I took a couple of slides from this organization. It's USAGE, I think that's how you pronounce it, or EU SAGE. It's a kind of a network of plant scientists, basically in Europe, and they are an important player in the discussion, and they are very much in favor of using CRISPR when it's for the best interest of society and for nature. I think that's a very good starting point to have a discussion like this. So I already mentioned that it's hundreds of cases have been reported in the literature now. So not only the tomato, but also rice and maize and soybean and some other very important crops worldwide. And so there is a lot of potential. So if we agree that we can use this technology, we can really speed up the improvement of crop features. And I think with the still growing world population and with the changing climate, this is really something that we should take seriously. Because in the Netherlands, I mean, there are very few people that go to bed with a hungry feeling. But in 2018 we had a CRISPR meeting in Wageningen, and that was just to talk about ethical issues. And then at some stage there was a discussion on stage by a guy from it was not Greenpeace, it was I think Save the World or so, an organization in England, and so he was talking about Monsanto and all the bad things that happened. But then the lady from Nigeria, she said, "Okay, now you have to stop because you should not be so arrogant, and you should give us the freedom to decide ourselves if we want to do this, and if we can use this tool to fight hunger, basically, then we definitely will do it. And I was very much impressed with that performance. Here you see a couple of examples of what has been done. So the percentage of the publications go down, but so much has been done to improve the nutritional value of certain crops. And then you can think about vitamin content and things like that. Also about the yield of the plants, or the fruit size and weight and things like that. So basically, how much are you producing? But also, this is a very important one: the stress tolerance. So the biological stress tolerance. So can plants defend themselves against attacks by bacteria or viruses or worms or whatever? Fungi, but also industrial utilization, so production of biofuels and things like that. So there are a lot of examples, and also a lot of examples that I think are in the best interest for society. An important thing an important detail in the whole discussion is this particular schedule. So it's kind of a risk assessment of editing. So how much of the DNA is in fact being changed. So this is so in red is the insertion of longer pieces of DNA, and one very or a well-known example is the introduction of a toxin gene, a toxin gene from a bacterium in mice. So it was the BT mice, and this toxin, of course, it made sense. This got the label of GMO, right? And that's perfect. That's how it should be, and because it's a toxin gene, so people theoretically could become sick of it, but that was not the case. And I guess the scientists that executed this work, they knew that from the beginning. This toxin was only a toxin for some beetles and animals, insects that were eating from crops. But if you feed the leftovers of those crops to cows, or if we would eat that mice, there is no problem at all. So that toxin is not harmful for us. But that is good to call that GMO, and that a GMO label kind of requires a lot of safety tests, and that's all good, right? But if you are talking about very small mutations, then I think that's not really needed, because many of those very small mutations, so you just break the DNA at a certain site to inactivate a gene. So you get those quick and dirty repair mechanisms, and then you introduce only a few letters at the DNA level that differ, and that also occurs in nature. So often those very simple CRISPR edits, you cannot even discriminate from what happens in nature. Of course, you can use it to speed up things. You can have a targeted knockout of certain genes. I just get a signal that I have to start thinking about wrapping up. This is an interesting map. It's not from Asterix and Obelix. This is about GMO, and in red you see the countries that call every CRISPR edit GMO, meaning this was a decision that was made by the European Court of Justice in 2018, and then people at Greenpeace were dancing on the table. Well, we won. But what they did not realize was that this decision was basically in the best interest of the big multinational companies, and not in the best interest for many smaller companies or even farmers. And if you also also the very small edits, what I already mentioned, only a few letters, they can They can be used to enhance the yield, to improve the resistance against disease, the tolerance against drought and salinity, so salt. So you can already, those very simple edits, you can use them for improving crops substantially. And that is what is allowed in the green countries. And we do not know what is going on in the orange countries, but I think you can figure out that also there the rules are less strict, at least than they are in Europe. So the good news is that there was a lot, and not in the least because of this EU SAGE organization. There was a big discussion started with the politicians in Brussels, and they are now reconsidering, and within a couple of months they come with a new decision. So let's see. Okay, so this is indicating that in Europe we are lagging behind, for instance, if you compare to China and the US, and that's for obvious reasons, because of the regulation. So my conclusions would be that edits should benefit society. That's the so-called Norway model. I think that is where it should start. When it's in the best interest for everybody, then I think it will be easier to convince the public that it's a good idea to do that. So it could be as simple as improving the nutritional value. It could also be to lower the price, for instance, things like that. Also, the small edits that I keep on talking about, they should not be called GMO, like already in the US and Australia and South America and Japan. Only the bigger edits should be called GMO, like transplanting a gene, as said. Then the Surprise Act. A few minutes. Stay with me. The patents, that's really also an issue. Because it's, of course, in the best interest for the companies again, but it's sometimes slowing down progress because not everybody has access to these patents. Also at Wageningen University we have a couple of patents, CRISPR related, and what we try to do, so we collaborate with a company in the Netherlands that use it in the biotech, so producing bioplastics, for instance. We spin off of our group, so a couple of students started a company, and they are developing diagnostics against COVID, for instance. So that's very interesting. I already mentioned the interaction we have with this group in Leiden to try to work on Duchenne. But in the plant field, we did not have any interaction yet. No company wanted a license. And of course, we only talked to companies in Europe. And for obvious reasons, they are very reluctant to start working on this. And then one day, it's I think 2 years ago, I was watching television, and then there was this colleague from Wageningen, Christa Testerink. She's professor of plant physiology in Wageningen, and she was in the VPRO in a program, and she was talking about what she was doing, and she explained that she was trying to work on the problem of salt tolerance in plants, but also drought resistance. And then she also said that she was interacting. So the next shot, in fact, of the program was that she was walking. It's not her, but this is somebody else. That she is also collaborating with this non-profit institute in the Philippines. And well, she tries to help these people. And then I thought, well, why don't we give away free licenses to these kind of non-profit organizations? Then I discussed this with the boss of our university, and she is in Louise Fresco, and she is, well, the university is the owner officially of the patents. But when I proposed this idea, she really was very enthusiastic about it, and this was launched during the opening of the academic year a year ago now, but now we have to make it work, right? And in fact, and that's the Surprise Act. We now have this Surprise project, and we want to optimize one of our enzymes. That is what. Oops. Did you do that? Okay. This is a sign from above. Then I have to wrap up. The good news is that we got we got in touch because of the publicity we have with the plan to provide free licenses. We came in touch with a wealthy person, an alumni from Wageningen University, and he's now supporting financially this project. And I sneak back. So we hope that this initiative, so we are now really going to do it. And we are going to, so the two groups in Wageningen are going to work together with the people from the International Rice Research Institute. And that's, we hope that it works, of course, that we can make edits to make better rice plants. But not only that, we also hope that it will kind of be a showcase that also other people's. I mean, there are thousands of CRISPR patents. That's really holding back the progress of all these beautiful developments that we could make. So let's hope that all together at some stage we will at least do our best to contribute to fighting hunger. And I'm not saying it only needs CRISPR. Of course, all the other strategies should continue, but I think it would really be helpful to include CRISPR. If you want to develop an opinion, because I guess you kind of figure out what my opinion on this discussion is, if you want to look it up yourself, these are starting points. So there is a YouTube film, and there is this document from an EU advisory committee. And there they give a lot of good arguments, pro and against, so you should have a look at it yourself. So my overall conclusion, so this is the last slide. So editing of humans, of course, for genetic disease, that's fine, but I wouldn't go any further. GMO classifications, in case of minor edits, not the way to go. The Norway model, I think, is very good, rather than the Monsanto model. So let's hope that contributes to convince more and more people. Also, I think, because this discussion between people pro and against editing and biotechnology, it doesn't make sense. So often they more or less have the same goals. So less water, fertilizer, herbicides, and energy, and more sustainable production of safe and healthy and tasty food. And I think that CRISPR can be a very nice role, and I think we should somehow get together and make this work. And about working together, this is the group of people, an excellent group of people that work with me, and we have a lot of collaborations also in other institutes. Thank you very much.